Greetings! Today, you will learn how to stain bacteria according to Gram technique, the most common staining method for differentiating gram-positive and gram-negative bacteria. First, we need to disinfect the slide, so we take alcohol and put it on paper. The next stage is application of a drop of physiological solution. But before we do this, it is necessary to light an alcohol burner, then take the physiological solution, open a test tube, flambe it, take a pipette, and apply a drop of physiological solution to the center of the slide. Next, we take an inoculation tube. In the same way as before, we disinfect it thermally by holding the alcohol bottle over the flame and spinning it from the edge. And then, at the last stage, we turn the loop vertically so that it turns completely red. Now we take microorganisms. In this case, we will be staining anthrax bacteria. We dip the loop and take the biomass. Now, we grind the biomass in a drop of physiological solution. We neutralize the biological material that remained at the tip of the loop. The next stage is the drying of the smear. All microorganisms are divided into gram-positive and gram-negative ones. Gram-positives have a thick layer of peptidoglycan. It occupies more than 80% of the cell wall. Gram-negatives have a thin layer of peptidoglycan, and that is why these two groups of microorganisms perceive stains differently. Using the same staining method, Gram in this case, we can differentiate these organisms by color, namely gram-positive and gram-negative. Now we need to fix them. Fixation performs two functions. Firstly, it sticks the smear to the slide, and secondly, it ensures the killing of microorganisms. We take the slide and quickly pass one side through the flame and then the other. After the glass has cooled down, we apply a filter paper that has been pre-moistened and dried with a gentian violet dye. Then we apply paper and fill it with water. Without washing the filter paper, we immediately apply Lugol's iodine. The drug should turn black. It is at this moment that gentian violet reacts with Lugol's iodine and penetrates the bacterial cell wall. We pour a little water to the dyes and apply alcohol. During this operation, it is necessary to shake the drug a little. What is the purpose of alcohol at this stage? If we have gram-positive bacteria, alcohol will cause the cell wall to swell and the complex of gentian violet and Lugol dyes will be retained in the cell structure. And if the bacteria are gram-negative, then alcohol will, on the contrary, wash out these dyes. We take water and wash the drug. At this stage, if we look under the microscope, we will see only gram-positive bacteria since they have retained the dyes. But what if we have gram-negative bacteria? Then they need to be stained with Fuchsin-Pfeiffer solution. We apply it to the center of our drug. Now, gram-positive bacteria will not be stained since their cell wall is already saturated with dyes. Gram-negative bacteria will be stained red, the color of fuchsin. Now we wash it again with water. We take the filter paper, dip the slide, dry it, and proceed to microscopy. So, you can see gram-positive anthrax bacteria, the purple rods, in the CSF sample. The gram-negative bacteria would be pink. Other cells are leukocytes.